Right. So we spoke about the pathology of insulin, but here we just want to focus on the pathogenesis of the different types. So pathogenesis is we are trying to look at how these disease conditions actually came to be. All right, like how did they actually come true? You understand? So for pathogenesis of type 2 diabetes mellitus, it's like this. Of course, diabetes mellitus type 2. This one, there's insulin, but there's resistance to insulin. You understand? So there's an interplay between genetic and environmental factors, you know, but there's no evidence of autoimmune basis, you know. So if you are looking at the genetic factors to developing type 2 diabetes mellitus, right, so the concordance rate of 90% in monozygote twins. So first degree relatives though, they have 5 to 10 folds higher rates of developing the disease than those without a family history, you know. So literally, if somebody has insulin, you also do like all those family history for them. Yeah, if somebody has diabetes mellitus, you have to do all those family history. Environmental factors, obesity. So more than 80% of those with type 2 diabetes mellitus are obese. So obesity is actually a factor here. So the important environmental factor is obesity. Then the other one is sedentary lifestyle. So you are not active. Metabolic syndrome. Okay. Sleep disorders such as obstructive sleep apnea and circadian disruption. So metabolic defects in type 2 diabetes mellitus is there's insulin resistance. So over time, the B cells are producing insulin, but they are not using, your body's not using insulin. So you have a backlog of insulin on the beta cells of the islets or pancreas. So for the insulin resistance, there's a failure of target tissues like liver, skeletal muscles, adipose tissues to respond to insulin. So there's functional defect in insulin signaling pathway. All right. So this is what underlies the insulin resistance. All right. So consequence of insulin resistance is, of course, you just have glucose whirling around the body with nobody to break it down. So it's going up. Postprandial blood glucose levels. So glucose do not enter skeletal tissue like postprandial. That's after you eat. So when you eat, glucose levels shoot up so high, you know. Excess triglyceride breakdown. Why? Because in adipose and free fatty acid, the body is trying to look for alternative levels, alternative energy, you know. So beta cell dysfunction is impaired glucose tolerance, increased beta cell functions in order to compensate. So this continues until their capacity to induce euglycemia fails. Then diabetes comes in. So mechanisms which promote B cell dysfunction include lipotoxicity, glucotoxicity, abnormal incretin defect, amyloid deposition in the islets, polymorphism in genes, controlling insulin release, right? Monogenic diabetes. So there are a group of rare forms of diabetes mellitus that result from mutations in a single gene. Right, so this one is now talking about mutations. Alright, so this usually results in what? From a primary defect in beta cell function or a defect in insulin receptor signaling. So you have mutations of what? Gluconeogenesis, you have mutations of RTK genes and all of that. Alright.